Okay, so let's go ahead and get started. First of all, CRAS, Cancer Research and Awareness Society, is today having a collection of cheek swabs between 10 and 2 in order to be deposited into a bone marrow registry for patients that have cancer, and in the future you may be able to donate white blood cells to those particular patients that have whole body radiation. So today, if you can, go between 10 and 2 today on, it's actually I think in front of the Student Union, and you'll be able to possibly save a life in the future. Today it's very easy. It's just a cheek swab. I think you just get like a Q-tip and wipe the side of your mouth. They collect the cells. They'll do the DNA analysis. They'll do something called HLA typing based on your DNA. And then if sometime in the future someone needs to have a bone marrow transplant, you could actually save a life. So look into it, and I highly encourage it. Don't forget there's going to be a quiz on Wednesday. The quiz will cover everything since the last midterm up through the end of today's lecture. So that will be the coverage of the quiz. It will be between 15-20 minutes in duration. It will likely not be multiple choice, so practice the skills that you've been doing in order to do the problems in the book, in order to catch up on the lectures that I've been giving, and also the sapling homework. So it will be based on one or more of those items. Last time I had talked about metabolic fates of pyruvate, and so we talked about how pyruvate can be converted into lactate. Okay, so I talked quite a bit about that, and I also talked about how pyruvate can be converted into ethanol, okay, in a different organism, that's yeast, and I'm going to talk today about how pyruvate can be converted into acetyl-CoA, okay? And this actually begins the what's known as the citric acid cycle, or the Krebs cycle. So from here, from acetyl-CoA, we'll go into the Krebs cycle. And by the way, I think you'll find this material in chapter 14. Okay. So what do we need to do? So pyruvate, if it's going to go to acetyl-CoA, and we do that for very efficient production of ATP, that pyruvate needs to move from the cytoplasm, where it's produced by glycolysis, into the mitochondria. So I'll just go ahead and draw this mitochondria. It has two membranes, an outer membrane and inner membrane, and then it also has this inside part called the matrix. Okay, so that's called the matrix. And so this pyruvate needs to get transported into the matrix in order for this particular reaction to occur. What is this reaction? What is it being carried out by? It's being carried out by pyruvate dehydrogenase complex. Okay, and from this name you could tell right away that's going to be an oxidoreductase reaction. And what you're going to see here is not so much the production of ATP directly, but now we're going to produce a lot of NADH, and that NADH will be used for oxidative phosphorylation. So, and oxidative phosphorylation is one of the major roles that the mitochondria carry out. Okay, how does it happen? So first of all, we start off with pyruvate. And pyruvate reacts with NAD+, and also something called coenzyme A, which we will abbreviate as CoASH, and then that's going to get converted into acetyl-CoA, which we'll abbreviate as, we'll just say acetyl-CoA, plus CO2 plus NADH plus a proton. So those are what's being made. Recall that pyruvate will look like this. I'll sort of draw it in a different way. So that's pyruvate. It's a little bit different than what I used to draw it as, but that's the same structure. And then acetyl-CoA, what does that look like? Acetyl-CoA will look like and we just abbreviate this as S-CoA. This is a cofactor, this coenzyme A, which is pretty large. Okay, let's talk a bit about the molecules involved in this pretty big pyruvate dehydrogenase complex. That's a very large set of enzymes that work together. They work in concert. They're actually three enzyme activities placed into one. Let me just first write down what those three enzyme activities are. So I'll just say pyruvate dehydrogenase complex. And what are those three enzymes? We know them as E1, E2, and E3. So those are the three components. And E1 is pyruvate dehydrogenase plus some cofactors, and the cofactor is thiamine pyrophosphate, which we also name as TPP. So that's just an abbreviation for thiamine pyrophosphate. Thiamine, by the way, is vitamin B1, and without thiamine, without that vitamin, we're unable to process. This enzyme activity is unable to go on. So a lot of these vitamins are these cofactors, as I keep on saying. Okay, E2. What's E2? E2 is dihydrolipoyl. Dihydrolipoyl transacetylase. Okay, and the cofactors for this enzyme are lipoamide, and coenzyme A. I'll just abbreviate that as CoA. And then what about the third enzyme? The third enzyme is known as dihydrolipoyl dehydrogenase. Dihydrolipoyl dehydrogenase. And the cofactors that are involved are flavin, adenine, dinucleotide, and we abbreviate that as FAD. Okay. And a component of this is called riboflavin. And so riboflavin is another one of these vitamins that you'll see in the back of cereal boxes if you guys eat cereal. Okay. And also nicotinamide, adenine, dinucleotide. And of course, that we've already talked about, that's NAD. So there's a lot of cofactors that are involved in this reaction. I want to just acquaint you with these cofactors. So we're going to turn to page 538. Page 538 of your book. Just to briefly acquaint ourselves with these cofactors. Here we go. So let's actually turn to page 537 of your book. And this has a picture of thiamine, which we ingest. Okay, so we actually take in thiamine from sources that are rich in vitamin B1. So B1 is thiamine. And eventually that thiamine, once it gets into our bodies and into our livers and into our other organs, it gets converted into thiamine pyrophosphate. And so thiamine pyrophosphate is created. And all that is a pyrophosphate from the ATP gets attached to this alcohol group. The pyrophosphate is coming from the beta and the gamma phosphates of ATP, and we're left with AMP. And that's a fairly easy mechanism that you guys should be able to know. What I've done here is I have circled the business end of thiamine, and so when I say the business end, this is where the reactions are going to take place. It actually takes place in this little area right here, okay? And specifically, that carbon is going to lose its proton, and it's going to become a good nucleophile. And so we're going to make what's called a carbanion. Carbanion, that's a carbon, that's an anion, right? So it's going to be negatively charged. And so that's going to be the key player in this. Okay, how does it work? Let's go ahead and okay, before I do that, I'm going to introduce you to the other cofactors. That's just one of the cofactors. Let's talk about the other cofactors. The other cofactors are found on page 599 of your book. So I'm going to kind of roll these out for you. And this is called lipoic acid. Lipoic acid gets attached to a lysine in enzyme number 2. So that lysine here, here's the side chain of lysine, and you can see it makes this linkage with this carbon. Any chemists here in the audience, can you guys tell me what type of linkage that is between this nitrogen and this carbon and this oxygen? Amide. That's an amide linkage. Very good. Amide linkage. So that's exactly right. And so we call this lipoamide, right? So you see that's a poamide. So that lipoamide is going to be linked here, but the business end of this is actually going to be this disulfide bond. So that disulfide bond is going to undergo reduction, and then oxidation, then reduction, then oxidation. So this is actually going to be really important. And that's actually shown here. You add 2 protons and 2 electrons, and you're going to basically break that disulfide bond. This is the reduced form of the lipoamide, and this is the oxidized form of lipoamide. Okay? All right. So that is the business end of the lipoamide, this disulfide bond. What else? What other cofactors are there involved? There's one which I've already talked about, this thing called FAD, flavin adenine dinucleotide. Okay? And the flavin adenine dinucleotide has this riboflavin, which again we ingest. We ingest the riboflavin, and that gets converted into something called FAD, flavin adenine dinucleotide. It's right here. And I'm going to circle the business end of this flavin adenine dinucleotide. It's this part right here, this part of the ring system, nitrogen, carbon, carbon, nitrogen. That's the business end of this whole thing. So concentrate on that when we start to go through the mechanism. And finally, there's something called coenzyme A. So here's coenzyme A. That is coenzyme A. And coenzyme A, the business end of the coenzyme A is the sulfhydryl group right here. So this is the part which will attach to the acetyl group. Okay, so that thiol or sulfhydryl group. That's what will happen. And we've already talked about NAD. Okay, so I think now we're ready to sort of talk about this very complicated but very important enzyme complex. And I'm going to turn your attention to a figure on page 602 of your book. And this is just to sort of give you an overview. We'll go into details in a bit, but this is the overview. Here is enzyme 1, enzyme 2. Enzyme 1 is in yellow, enzyme 2 is in green, and enzyme 3 is in this is pink, okay, or red. And so here we have pyruvate coming in, CO2 is going to leave, and then this enzyme 1, which has thymine pyrophosphate, is going to transfer this acetyl group to lipoamide. So it's going to get attached to lipoamide. So here's lipoamide, and that's on enzyme number 2. And then the lipoamide then goes ahead and transfers the acetyl group onto coenzyme A. And so here's the other product. Here's the product, acetyl-CoA here. And then we've got this reduced sulfur. Okay, and that reduced sulfur then is going to finally get oxidized. So it's right here. This is the reduced sulfur here. And it's going to get oxidized. And that's what enzyme number 3 does. It does all the oxidation. First, what it does is the electrons here are going to get transferred from the reduced lipoamide onto FAD. And then the FAD is going to transfer it onto NAD+, and then NAD+ is going to be reduced to NADH. The goal here is to basically capture the electrons in NADH. Okay. The handout that I gave you, you guys didn't get the handout, you come on up and get it, but the handout that I gave you is from another book, which I think illustrates this in a little bit clearer light. So you guys are welcome to come on up and get it if you didn't get it. Okay, so remember, the goal here is basically to take off the carboxyl group from pyruvate. The goal is to take off the carboxyl group from pyruvate. So here we've got the thymine pyrophosphate, okay, and the thymine pyrophosphate, as I said before, this is the business end right here. They just have R groups for all the rest of it, okay, and a base from the enzyme is going to remove a proton, okay, and now you've got this carbanion that I talked about, so that's the carbanion. And that's going to be a good electrophile, and this acid, this ketone carbon right here is going to be a good It's going to be. I'm sorry. This is going to be a good electrophile. The carbanion is a good nucleophile, and eventually, what happens is that you create an alcohol right here, and the enzyme again provides an acid. So actually this is an acid. It's going to provide this proton here. And then eventually you see the electrons here in the carboxyl group is going to feed into the carbon, and then now you get CO2 to leave. Okay? And the electrons here are between the carbon-carbon bond in the pyruvate is going to get broken. And so now we create this sort of resonance-stabilized carbanion. Okay? Key is this group right here. So just remember this group right here. You don't have to remember this. Okay? And this negatively charged carbon is going to attach to the oxidized lipoamide. So this is the oxidized lipoamide. And the disulfide bond is going to break, and a proton is going to be picked up again from the enzyme. And so then a base is going to come along and attack the alcohol, and you're going to then have TPP leave. So TPP is going to leave, and now you've got this acetyl group that's attached to the lipoamide. Then we have coenzyme A that has a couple of lone pairs of electrons on the sulfur of coenzyme A, on the sulfhydryl group. That's going to attack the carbonyl carbon here, and then you get acetyl-CoA, and then the lipoamide is now in the reduced state. So we have the reduced state right here. Okay. The rest of the enzyme activity is really just to capture those electrons that are there in the lipoamide and place them into NAD plus to make NADH. What is the next step in this reaction? Okay. So we've got this, I'm on page 603 of your book. Okay, so this is 603 of your book. Here I left you with this reduced lipoamide. So this is the reduced lipoamide. And then we have this sort of disulfide bond that's inside the inside the enzyme. And so we get reduction of that disulfide bond. And eventually, the electrons that are in this thiolate are going to attack FAD. Remember I told you that for FAD, this is the business end of the molecule. So it attacks this carbon that's what's attached, that has a double bond to the nitrogen. And oh, by the way, the lipoamide is recreated here, so that we got this oxidized lipoamide here. And then everything happens on FAD. So here's FAD, and then the disulfide bond is recreated inside the enzyme, and then we have a concerted reaction where you have these electrons are going to create this double bond within this ring, this ring system. And then you get protonation of this nitrogen. And then a base comes along, removes this proton, and then you get this concerted reaction again, where you get movement of these double bonds, and eventually you get this negatively charged hydrogen. Negatively charged hydrogen. Do you guys remember what that's called? Hydride. Very good. Hydride. And the hydride will attack NAD+, and I've already gone through this mechanism, and NAD+ will get reduced to NADH. Okay? And it's this NADH, as I said before, which is the final cofactor that's reduced that comes out of this. By the way, I want to have you guys write this down. FAD is kind of interesting because it can donate either a hydrogen, that means basically an atom, a proton with a single electron, or it can donate a hydride. And it just depends on the reaction that it's in. So just make a note of that, because we'll go through another reaction where FADH2 is going to donate a hydrogen instead of a proton. Or maybe, no, actually it's the opposite. A hydrogen gets added to FAD to make FADH2. So I'll show you that mechanism when we get to the Krebs cycle. Okay, so this is the major mechanism here, and so we actually have now completed a number of the metabolic fates of pyruvate. We've talked about how it can get converted into ethanol. We've talked about how it can convert to lactate. We've talked about how it can get converted into acetyl-CoA. And so now I'm looking for my special pen here. Oh, there it is. Okay. All right. Are there any questions on this? Make sense? Okay. Good. Okay. I want to talk a little bit about what's called the Cori cycle. I want to step back and talk about the Cori cycle. Haven't talked about this yet. What is the Cori cycle? Well, remember I was talking about last lecture how you can make lactic acid if you're doing strenuous activity, muscle activity. And that lactic acid can get eventually exported from the organ into the bloodstream, and how that makes the pH of the bloodstream go down, and that has a Bohr effect on the hemoglobin. So we talked about that. Well, eventually that lactic acid, we would like for it to be productive, and so we can actually make glucose out of that. And we can make that glucose through something called gluconeogenesis. And a major organ that does gluconeogenesis is the liver. Okay? So the liver does a lot of this work. And then the liver can make glucose and actually export that glucose back into the blood, send it back to the muscles, and the muscles can then use that glucose for more energy production. So on page 547 of your book, okay, I'm going to show you this Cori cycle. This is from your book, page 547, named after the couple, the scientist couple that made this. So here we have the skeletal muscle. We'll start right here. By the way, glycogen, which is a polymerized form of glucose, gets broken down into glucose. I haven't talked about that, but there's a number of reactions that happens. But this is the stored form of glycogen. We don't store that much glycogen in our bodies. Only enough for about 20 to 30 minutes of strenuous activity. So that glycogen gets broken down into glucose. We've talked about how the glucose, you can make a net of 2 ATP molecules. We've talked about that. You guys have in your minds how those ATP molecules are created through substrate level phosphorylation, right? And then there's actually a little bit of a mistake here, so I'm going to write this in here. And then 2, of course, 2 also NAD pluses get converted into 2 NADHs. Okay, and that happens at the level of glyceraldehyde 3-phosphate dehydrogenase. Okay, and that's how we get this NADH out of this. And then 2 pyruvates are made. Okay, this all happens in the cytoplasm. Okay, and then in order to quickly make more of this NAD+, because I told you that NAD+ is limiting. There's not very many of those molecules. Maybe they're complicated to make. Maybe we don't have the capacity to make a lot, but we don't have very much of that NAD+. So we gotta remake this NAD+ so it can be used for more glycolysis, right? When you're running in order to catch your bus or whatever it is, okay, you gotta make more of this NAD+. Okay, so basically these 2 NADHs come over here, and that's the 2 NADH. Okay, so now we've made 2 lactates. Okay, the lactates, as I said, goes into the blood, lowers the pH of the blood. That causes you to breathe more heavily. Okay, and then the 2 lactates get transported into the liver, and in the liver, the lactates can actually be converted back into pyruvate. Okay, Okay, so we get this pyruvate going back here. And then through a process known as gluconeogenesis. It's a good name, right? Glucose for glucose, and then, genesis means to create, I guess. Okay, so we're making it again. We're making new glucose from this. Okay, so we get actually 2 of these NAD pluses are going to come in here and make 2 NADHs. We get 2 pyruvates, and then these 2 pyruvates can actually be sent back to glucose. Okay, and this requires. It says here this requires 6 ATPs. I'm going to have to check that. But anyway, this requires some ATPs, and you make glucose, and the liver then can put this back out in the blood. The liver is the organ that's like the master regulator of glucose in your blood. Okay? So it's like responsible for like checking to make sure that blood has enough glucose to supply muscles and other organs with the right amount of glucose. So that's why the liver is hormonally regulated by a couple of different hormones, right? One is insulin. Insulin will regulate glucose metabolism by binding to receptors on the liver cells, and also another one's called glucagon. When you're starved, glucagon comes into play. Glucagon will bind to the liver, and when you've just had a meal, just had a wonderful breakfast at Bob's Big Boy restaurant, okay, and you've had omelets and you've had hash browns, then the insulin kicks in and causes the liver to sort of take in glucose, and other organs take in glucose. So that's where those take place. Yes?
What about epinephrine?
Yes, epinephrine is a third hormone that comes into play as well. So epinephrine is somewhat like glucagon in that it will provide glucose. It will provide an increase in glycolysis, especially in muscles. Right. Okay. But it's just hard to think about all the hormones. Okay. Right. But epinephrine is another one. Okay. So we've got these systems here, and I want to talk a bit about this thing called gluconeogenesis. This gluconeogenesis. How does it work? Your book actually does a pretty good job on this. Here we go. Okay. So on page 542 of your book, on the left-hand side here, you've got glycolysis on the left, all these arrows going from glucose down to pyruvate, and you've got gluconeogenesis on the right. A lot of the same enzymes are used for glycolysis and gluconeogenesis, but not all of the same enzymes, but most of them, okay? I'm going to point out the differences. Actually, your book does a good job here. Any thick arrow here uses a different enzyme to go in the reverse direction. Let's just first talk about glycolysis, since we're familiar with this. So here we have a major enzyme called hexokinase that goes from glucose to glucose 6-phosphate. I talked about this major enzyme, phosphofructokinase. Okay, so that's this enzyme right here that goes from fructose 6-phosphate to fructose 1,6-bisphosphate. And there's a couple of enzymes here that also are important, from going from phospholipyruvate to pyruvate. To pyruvate, that is pyruvate kinase. So these 3 enzymes are really important, and they're regulated. They have allosteric sites that regulate them. So they respond to energy charge. And you guys know how the energy charge would work to either turn off or turn on these enzymes. Energy charge. Allosteric sites. Enzymes. Okay. What about going in the reverse direction? In the reverse direction, you've got a couple of different enzymes here. Pyruvate carboxylase and PEPCK, phospholipyruvate carboxykinase. Okay, those are the enzymes here that are different. then all of these enzymes here with these little tiny arrows, they're the exact same enzymes that you use for glycolysis. Okay? Then you got fructose 1,6-bisphosphatase here, and then you got glucose 6-phosphatase. So these are the enzymes that are being used for gluconeogenesis. One thing I want to say off the bat, you don't have these things going on at the same time. It's not like, the 710 freeway where you've got, trucks going in one direction, you got trucks going in the other direction at the same time. No way. The reason for that is that we would cause something called a futile cycle if we did that. Futile cycle. Okay? And that's bad news. Why is it bad news? It's bad news because basically you'd just be wasting energy. You only get 2 ATP molecules for glucose going to pyruvate. You only get 2 ATP molecules. However, you get a total of 4 ATP. You consume a total of 4 ATP molecules and 2 GTP molecules going in the opposite direction. You're going to be down 4 ATP molecules every single time you do this futile cycle. The reasons why we have these different enzymes here, and they're allosterically regulated, is because gluconeogenesis and glycolysis respond exquisitely to energy charge. When energy charge is high, then you have gluconeogenesis. Okay? When you have energy charge that's low, then you've got glycolysis. Okay? So they do not run at the same time. Okay, let's talk about details about gluconeogenesis. There are what we call 3 bypasses. Why are they called bypasses? Because basically we're using different enzymes. This is called bypass number 1. I'll just underline it here. Bypass number 1. This whole thing is bypass number 1, going from pyruvate to phosphoenolpyruvate. What's bypass number 2? Bypass number 2 is this one, and bypass number 3 is this one. So those are the 3 bypasses. They're called bypasses because we're not using the same enzymes as we do for glycolysis. That's why they're called bypasses. Let's talk a little bit about these bypasses. We've talked about metabolic fates of pyruvate. I've talked about how pyruvate can get converted into ethanol, how it can get converted, well, that's not in humans, okay, that's in yeast, okay. I've talked about how pyruvate can get converted to lactic acid. I've talked about how pyruvate can get converted into acetyl-CoA. Just went over that today. There's a fourth place where pyruvate can go to, and that's called oxaloacetate. Oxaloacetate. So here's pyruvate again, okay. We've been hitting on this molecule quite a bit. It can react with bicarbonate and ATP, and we're going to create oxaloacetate, and this requires some ATP. Okay. Next, this is all part of bypass number 1. Oxaloacetate reacts with GTP, and we create phosphoenolpyruvate. So that's the end of bypass number 1. So 2 reactions. Pyruvate going to oxaloacetate, which, by the way, this requires a cofactor known as biotin. Okay. And then we've got oxaloacetate plus GTP going to phosphoenolpyruvate. And this is a pretty simple reaction. Here we've got, let me see if I can just focus in on this so you guys can see it really well. There we go. Okay. Here we've got GTP. Actually, this is GDP with a terminal phosphate, the gamma phosphate. Here we have the carboxylic acid on oxaloacetate, and you got this concerted reaction. Yeah, boom, boom. Okay, and then this third reaction where you take off the phosphate, and so now you got the phosphate attached to this guy. You got CO2 leaving. This is CO2 leaving, and what we've created is phosphoenolpyruvate. This is phosphoenolpyruvate. So that is the reaction here. We're going to stop here on bypass number 1. Bypass number 2 and bypass number 3 are very simple. You can do those in your sleep. Okay. Quiz on Wednesday.